So in this video, we're going to talk about a decarboxylase test, and we're going to start with day one, which is the setup of this test. And this can be found in your lab book with activity 5-11. And so let's talk about how we would set this test up. So the purpose of the decarboxylase test is to determine whether bacteria can decarboxylate lysine or ornithine. Lysine and ornithine are amino acids. And if you remember in our chemistry lecture, when we talked about amino acids, they have this alpha carbon, they have an amino group, they have a carboxyl group, they have a hydrogen, and they have an R group. And remember that the R group is the variable portion of the amino acid. That's the part that's going to vary between lysine and ornithine. And so when we talk about decarboxylation, we're talking about removing this carboxyl group. In the next video, we will talk about deamination, which is the removal of the amino group. But in this test, we're looking at the decarboxylation, meaning to remove that carboxyl group off of the amino acids. So this experiment would be done normally in groups of 4. And the way that this would work is you would have 2 red cap tubes, and the 2 red cap tubes are the ones that would contain lysine. And lysine is our amino acid. So the red cap is going to have lysine, the blue cap is going to have ornithine. So what we would do is we would, for each organism, we have 2 organisms total. We have Enterobacter aerogenes and we have Proteus vulgaris. So if I was in charge of Enterobacter aerogenes, I would inoculate both a red cap and a blue cap with my EA. So I would use a loop and my aseptic technique and pick up bacteria and transfer it to 2 tubes, one with the red cap, one with the blue cap. Another member of my group would do the same thing, but this time using Proteus vulgaris into a red cap and a blue cap. Notice that after we inoculate the broth, we would layer them with mineral oil. Now, if you think back to our OF test, our oxidation and fermentation test, what is the purpose of putting a layer of mineral oil? And the answer is that the function of the mineral oil is to make the tube anaerobic, meaning to seal out the oxygen. So what that tells you is that this reaction is anaerobic. It happens in the absence of oxygen. And so I have a video demonstrating how to do the setup for just one of the organisms. And so here's the video showing you the setup. So in this video, we are going to set up our decarboxylase experiment. And so what we have is we have 2 types of decarboxylase broth. We have an ornithine decarboxylase, which is the one with the red cap, and we have a lysine decarboxylase with a blue cap. The difference between them is the amino acid substrate that's present in them. And when we do this experiment, we have 2 bacteria that we will be using. We have Proteus vulgaris. You can see it growing on the slant. We also have Enterobacter aerogenes, which again is also growing on the slant. And so let me demonstrate how we're going to set this up. So notice that I have 2 red caps and 2 blue caps. So what that means is for each organism, I'm going to inoculate a red and a blue. So like these would be PV, these would be EA. When I do the demonstration, I'm only going to demonstrate one, just so you get the idea. So what I need to do is I need to start with my loop, and I'm going to flame sterilize my loop. Starting at the base, working towards the tip. I'm going to let that cool. While I'm letting that cool, I'm going to do EA. So I'm going to twist the cap loose. And I'm letting my loop cool. And then I'm going to take my tube, hand up, palm up, put the cap between my pinky and ring finger. Flame it. Go in. Pick up some bacteria. Flame it. Cap back on. I'm going to inoculate the red cap first. Flame it. Add it in the broth. Flame, cap. I'm going to move it in my rack so that I know that that tube is already done. And then I'm going to reflame sterilize. And I'm going to let it cool. I'm going to repeat the same thing with the blue cap tube. Same organism. That organism is going into both a red cap and a blue cap. So I let it cool. Take the cap between my pinky and ring finger. Flame it. Go in. Pick up some bacteria. Flame. Cap. Set it down. To the blue cap. Flame it. Swirl it in. Flame it. Cap. There's liquid on my loop, so I have to be careful. And I'm going to flame from the base, working towards the tip. And again, there was liquid on there, so I want to be sure to do that well. So I'm done with my loop. Now, this test is anaerobic. So just like when we did our OF test, how we had an aerobic tube and an anaerobic tube, this test is anaerobic, which means that we need to take these tubes that I've inoculated, and to make them anaerobic, I need to add mineral oil. So I'm going to add a layer of mineral oil to both tubes, just enough to seal out the oxygen. So I'm going to loosen the cap. I'm going to take this, flame it, and then I'm just going to hover my dropper over the top. Flame it, cap back on. So notice I have a nice little layer of mineral oil on this tube. That's going to seal out the oxygen. So I need to repeat that for the blue cap. Flame it. You don't want to contaminate your dropper. Flame it, cap. And again, I have a nice little layer of mineral oil. So those tubes are done and they're ready to go in the incubator. And then I would repeat the same thing with my other organism. And so this is going to be your decarboxylase experiment. Okay, so now we're going to talk about our decarboxylase test, day 2, how we would do our readout. And this readout is going to be done after 48 hours after inoculation. So before we can do that, we need to talk about what is in the media. So again, we are looking in this activity, in this unit, we're starting to really focus on how bacteria metabolize proteins. In the last unit, we were looking at how bacteria use sugars. And so we did an ONPG test, and we did a phenyl red test, and an OF test, etc. Those tests were looking at how bacteria utilize sugar. In this experiment and in this unit, we're looking at now protein metabolism. We're looking at how do bacteria metabolize protein as a food source. So just like for the sugar test that we were doing, this is also going to use a biochemical sheet. So you will be filling out a biochemical sheet for your decarboxylase test to help you to study. And so the name of the media is going to be our decarboxylase media. That's the name of the media. So just decarboxylase media. In the media, we have peptones and beef extract. What are peptones and beef extract in the media for? And the answer is the peptones and the beef extract, right? Food for general growth. It's there just to allow the bacteria to grow. It's just there in this case for general growth. So what that tells you is that this media is complex or undefined media. It has ingredients that can vary from batch to batch. Both the peptones and the beef extract will make this media undefined or complex media. And so that's the first ingredient. The next ingredient in the media is going to be the glucose. Now, remember I said in this unit that we are looking at the metabolism of protein. And so you might be wondering, well, why do we have glucose in the media? Well, the glucose is there for fermentation. Remember I told you that bacteria, if given the choice between glucose and protein, bacteria would choose to use the glucose first. However, in this media, the glucose is there only at a low concentration. And the reason is that we want the bacteria to utilize the glucose first because when bacteria metabolize sugars, remember that the products are going to be acids. Those acids are going to lower the pH. In order for decarboxylation to occur, there needs to be a low pH. So the glucose is there basically to allow bacteria to ferment the glucose. That will help drop the pH or decrease the pH, which can then allow decarboxylation to occur. So that's why we have glucose in the media. Next, we have our bromocresol purple. Now notice the name. What do you think bromocresol purple is? Well, it's a pH indicator. And remember that I talked about that there's a general rule when we talk about our pH indicators, and the only exception to that was methyl red. So what color would you expect bromocresol purple to be when it's acidic? Answer is yellow is acidic, right? Because I said for every pH indicator that we use, except methyl red, everything else, yellow is acidic. That's true for bromocresol purple. That is true for bromothymol blue. That is true for phenyl red. So yellow is acidic. Now, let's think on the opposite end of that spectrum. What color would the alkaline be? Well, again, there's another rule. Remember I said, notice the name is bromocresol purple. That second color, that color that's in the name, that's the alkaline. So the purple is the alkaline. Now, if you mix yellow and purple together, so the neutral color is kind of this orange color. So if it's kind of orangey, that is going to be our neutral. That is our starting color. That is the pH when we begin. Because remember that most bacteria are neutrophiles. They grow best at a neutral pH. So that kind of orangish color is going to be our neutral. And again, purple is going to be our alkaline. The name tells you the alkaline color. Next, we have our pyridoxal. And pyridoxal is vitamin B6, which is a required coenzyme. And what that means is that this vitamin B6, this pyridoxal, is required in order for the decarboxylase enzyme to be active. So many times enzymes will need these coenzymes or these cofactors that help the enzyme to function. And so in this case, decarboxylase, that enzyme, needs this pyridoxal. Without this coenzyme, the enzyme is not active. And so we have pyridoxal in there as a required coenzyme. Lastly, we have our L-lysine or our L-ornithine. Those are our amino acids. So those are going to be our amino acid substrates. And remember that when we talked about amino acids, that the L isoform is the one that's more common in nature. And so that's the one we're going to use in this test. We're going to use the amino acid isomer that is more common in nature. And so in our tubes, one tube had lysine, the other tube has ornithine. So they're not mixed together, it's or. One has lysine, one has ornithine. So this is our amino acid substrate for our test. So if we look at our reaction for the decarboxylase. So again, in order for this reaction to even occur, bacteria need to ferment the glucose first. And they need to ferment the glucose to lower the pH for the decarboxylase enzyme to be active. So when bacteria use the glucose and they ferment it, remember that that's going to allow them to produce acids, alcohol, and gas. And so when those acids are produced, that is going to lower the pH. And so if you were to look at these tubes at about 2 hours after incubation, the tube should be yellow. Remember that the yellow is acidic. So initially, your tube is going to look yellow because the bacteria would ferment the sugar. And when they ferment the sugar, they're going to produce the acids. Bacteria need to have those acids for the reaction to occur. So acids need to be produced. Then we can start to see decarboxylation. So our substrate in this is going to be our amino acid. And again, it's going to be either lysine or ornithine. So we have two substrates in this test. One tube has lysine, one tube has ornithine. Those are our substrates. What we're looking at in this experiment is the decarboxylation. And remember, that means to remove this carboxyl group. So our enzyme that is going to do this is referred to as our decarboxylase. Notice that our substrate is an amino acid, which is relatively small. So would you predict that decarboxylase is going to be an exoenzyme or an endoenzyme? And the answer is it's going to be an endoenzyme because the substrate is small, the amino acid can get into the cell, and so that enzyme is inside the cell to do the decarboxylation. So decarboxylase is going to be an endoenzyme. Now, in order for this reaction to occur, we have to have 3 requirements. So enzymes, you're going to learn, enzymes often are only active under certain conditions. And that's used for the cell to basically conserve energy, meaning they don't want to produce these enzymes and have these enzymes active all the time. They only want to be on under certain conditions if the bacteria needs them. So in order for decarboxylase to be active, we need 3 requirements. 1, the low pH. And again, the way that we get the low pH is by putting glucose in the media, and the bacteria then need to ferment the glucose to produce the acids to allow the reaction to occur. So now, thinking question, can you do this test the way it's designed on an organism that is non-saccharolytic, meaning one that doesn't use sugar? And the answer is no. The bacteria have to be able to ferment the glucose in order to decrease the pH for the decarboxylase to even be active. So if you're using an organism that is non-saccharolytic, this test is not likely to work. Or if you forgot to add glucose, this test would not work because you need those acids to be produced in order for the decarboxylase to be active. So one requirement is that we have a low pH. The second requirement is the pyridoxal. And so in the media, it's pyridoxal phosphate. And again, that's a required coenzyme. It's needed for the enzyme to be active. The last requirement, notice that these tubes have mineral oil. What does that tell you about their oxygen requirement? And what that tells you is that this reaction is going to be anaerobic. It happens in the absence of oxygen. If you were to forget the mineral oil, this test would not work properly. So we need it to be anaerobic in order for the reaction to occur. So those 3 requirements have to be met in order for the enzyme to be active. And so if bacteria produce decarboxylase, they're going to take the amino acid substrate, they're going to remove the carboxyl group, and our products are going to be what we call a diamine. You're going to see that this lysine and ornithine, the R group has an amino group on it as well. So we get these diamines and we get carbon dioxide, CO2. So those are our 2 products. We get our diamine, and the diamine, those amino groups, those are going to raise the pH, meaning it's going to make the pH more alkaline. The pH is going to go up. So our products, again, are going to be our diamines, and depending on our substrate, you're going to get a different product. So for lysine, the product is going to be cadaverine. For ornithine, the product is going to be called putrescine. And you're going to see this in the next slide, and I'll talk more about this in a minute. But the products are going to be the cadaverine or the putrescine. They are diamines, and we're also going to get carbon dioxide, CO2. So notice that if this reaction occurs, the products, those diamines, are going to raise the pH. So which color is going to be our positive in this test? Is it going to be the purple or the yellow? Which color is going to be our positive? Well, let's start with the purple. What is the pH like in the tube that is purple? And what that tells us is that the pH is alkaline, Remember bromocresol purple? Purple is alkaline. So the purple tube has the alkaline pH. What that tells us is that bacteria decarboxylated the amino acid and they produced the diamine, which raised the pH. So is that a positive or a negative for decarboxylase? And the answer is that is a positive. So my positive test is going to be purple with turbidity. Bacteria grew, they decarboxylated the amino acid, and they produced the diamine, which raised the pH. That's our positive. So purple is going to be our positive. So now let's talk about what the yellow color tells us. Yellow is going to be acidic. So the pH in that case is going to be acidic. And so if our pH is acidic, what did the bacteria use as a food source? Well, if they produce acids, that means they metabolize the sugar. So the bacteria in this case metabolized the sugar and produced acids, which lowered the pH. So is that positive or negative for decarboxylase? And the answer is that would be negative, right? Because if decarboxylation occurred, then we would get the diamines, which would produce alkaline products. But if we have acids produced, glucose was fermented, it produced the acids, but that bacteria was not able to do the decarboxylation reaction and produce those diamines. So this would be a negative test. So in this test, our purple is our positive, PP, purple positive. Yellow is going to be our negative. And again, remember, this is going to be opposite to what we looked at when we looked at sugar metabolism. We were looking for the acidic products. In this case, we're now going to start to see these alkaline products. So if we look at lysine and ornithine specifically, if we look at these amino acids, again, here is our alpha carbon, here is the amino group, here's the carboxyl group. This is the R group. If I compare the R group for lysine and ornithine, notice that the only difference between them is one of these methyl groups, the CH2 groups. So notice that this one is 3 carbons and then an amino group. This one is 4 carbons and an amino group. So that is what makes lysine and ornithine different. Their R group is different by 1 CH2. So our lysine is our substrate. That's our amino acid substrate. And in our decarboxylase reaction, we're going to remove this carboxyl group. And that is facilitated, the specific enzyme for lysine, the specific enzyme is called lysine decarboxylase. And you'll see that abbreviated LDC. LDC stands for lysine decarboxylase. That's the name of that enzyme. That is the enzyme. That enzyme is specific and it decarboxylates lysine specifically. It does not necessarily work against ornithine. It's very specific to lysine. Again, my lysine decarboxylase would require the pyridoxal. That's the required coenzyme. We would need to have the conditions be acidic and we would need it to be anaerobic. Now, if lysine is decarboxylated, if this carboxyl group is removed, we get CO2. So a product is going to be carbon dioxide, and our diamine in this case, notice here's the amino group, here's the amino group. So my diamine in this case is referred to as cadaverine. Now, cadaverine, what does that make you think of when you see cadaverine? And the answer is cadavers or dead bodies, right? And so this cadaverine produces, this diamine has a very putrid smell. If you think about like dead bodies and rotting and the smell that they give off from these diamines, think about why that is. So you have to think about that in your body, you have bacteria, right? Bacteria can utilize multiple food sources. When you die, right? When a person dies, their heart stops pumping, so they're no longer circulating blood, which means they're also not circulating oxygen. So the tissues become anaerobic. They're no longer getting oxygen. So the tissues inside are becoming anaerobic because oxygen is not being pumped by the bloodstream. So the conditions become anaerobic. The bacteria are inside the body. They need a food source. So what happens is they start doing decarboxylation of the proteins that are in the body. And when they do that, right, when they decarboxylate those proteins and they remove the carboxyl group for the amino acids, in the case of lysine, it's going to produce the cadaverine. The cadaverine is going to produce that really putrid, gross, rotting smell. And the carbon dioxide is what causes the body to bloat, right? If you've ever watched like crime shows, you see like bodies like float. Well, the bodies float because the bacteria that's in there, those bacteria will do this decarboxylase reaction because it's now anaerobic, and they will produce carbon dioxide gas, which causes that body to bloat. And so this reaction is what causes that putrid smell as well as that bloating of a dead body, because it's the bacteria inside that are doing the decarboxylation of the proteins. Now, if we were to look at ornithine, ornithine also has its carboxyl group. So here's its carboxyl group. The enzyme that's going to do this reaction is called ornithine decarboxylase. And this is abbreviated ODC. So you'll see it abbreviated ODC. And this enzyme is going to specifically decarboxylate ornithine. And so it's going to remove the carboxyl group, and the remaining diamine is called putrescine. So putrescine, think again, putrid smell. It's a very foul smelling diamine. So when you're studying this, you do need to know which substrate goes with which product. So lysine goes with cadaverine, ornithine goes with putrescine. For me, the way that I remember which one goes with which, O and P are closer in the alphabet, so O and P go together. So ornithine goes with putrescine, lysine goes with cadaverine. So lysine and ornithine are going to be your substrates, and your products are then going to be cadaverine or putrescine, depending on which substrate you started with. And so this is going to be your decarboxylase test. This is what we're looking for. So if we look at the organisms that are tested, so on the left, we have our uninoculated control. Notice that this tube is that kind of orangish color. It starts at a neutral pH. If we look at the two tubes that were inoculated with Enterobacter aerogenes, EA, the tube on the left has lysine as its substrate. The tube on the right has ornithine as its substrate. Notice that for Enterobacter aerogenes, we have purple, purple. What does that tell you about if Enterobacter aerogenes, do they decarboxylate lysine? Is that a positive or a negative? And the answer is, well, it's positive because it's purple. Remember, purple is alkaline, which is going to be positive in this test. For ornithine, it is also purple, so it's also positive. So in this case, Enterobacter aerogenes can decarboxylate both lysine and ornithine. Now, it's important to note that not every organism is going to be like this. It's not an all or nothing thing. Some bacteria might only utilize lysine. Some might only utilize ornithine. Some do both, like EA, or look at Proteus vulgaris. Proteus vulgaris, or neither turn purple. Lysine is going to be yellow. Ornithine is going to be yellow. That is going to be negative for decarboxylase and also negative for ornithine decarboxylase. So Proteus vulgaris didn't decarboxylate either amino acid. It's negative for decarboxylation of lysine. It is negative for decarboxylation of ornithine as well. So it did not decarboxylate either. And again, it doesn't have to be that way. In some cases, some bacteria might use one and not the other. When you are studying this, you do not have to memorize the result for EA and for PV. I don't care that you memorize what their result was. However, if given tubes, you should be able to analyze those tubes and tell me what it means, right? And so if we see the purple, it tells us that the pH is alkaline. pH had gone up, which means that bacteria did decarboxylate the amino acid because it produced the diamine. The diamine raised the pH, which caused the bromocressyl purple to turn purple. That's our positive. In the yellow is going to be the acidic. And if the pH is acidic, that means that bacteria used glucose as a food source. And if they use glucose as a food source, then that means that they're going to produce acids that will lower the pH and will cause the bromocressyl purple to turn purple, which is going to be our negative for our decarboxylase. And so this is our decarboxylase test.